Hello, everyone.
Right.
So it is my pleasure to introduce Professor Ko Sakai, who received his degree in bioengineering from the University of Pennsylvania. And after that, he joined Professor Amari's lab became BSI, and he later moved to the University of Tsukuba, and where he served for many years and was appointed Professor Emeritus last year. His primary field of expertise is vision science, covering both cognitive and computational neuroscience. In addition to his academic career, he has recently served as a lecturer at the Kakenhi Clinic at OIST, and where he shared practical knowledge of the Japanese research funding system. Also, he has valuable insights into international research environments gained through his many sabbatical stays abroad. And he has been my long-term collaborator and a trusted advisor and a senior colleague whom I deeply respect and continuously learn from. So today he will give us a lecture. The title is Seeing the World, Building Scenes from Pixels in the Mid-Level Visual Pathways. So please join me in welcoming Professor Ko Sakai.
Thank you, Kako. I am Ko Sakai from University of Tsukuba, which is located about 1 hour northeast of Tokyo. And first of all, I'd like to thank to everyone at the TSVP who made it possible that my visit to here. And I'd like to also thank to scientists at OIST who are open for collaboration and discussions, including Dr. Kenji Doya, Yukako Yamane, Tomoki Fukai, and others. And today, I'd like to talk about our study about understanding how we see the world. Well, actually, this slide is no good. So let me move this a little bit. Okay, now you can see the title clearly. And actually, this text has been here from the beginning, but this structure made it difficult to see the text. And actually, this is what's happening in the real day life. In the real world, scenes are full of objects and many occlusions. Yet somehow we can see what they are. So today I'd like to talk about why we can do that. So let's begin with some old pictures. So this is a famous painting by Raphael. We can clearly see the 3D structure around the ceiling, staircase, people standing, those in the front, and others. But what you're actually looking at is this two-dimensional screen. Physically, there is no information about the 3D. And a few hundred years later, Monet painted this picture. So here we can see a tree, garden, house, a child, and probably her sister, and many flowers. But we move in to this flower. Actually, what we can see is only the red and white and probably green paint. So we don't see a flower here. So the point here is the seeing is different from measuring physical properties. It depends on how the brain constructs it. And some years later, the Picasso painted this picture. And even in his style, we can see here's a woman sitting on a red chair. Again, the point is, seeing is different from measuring the physical properties. So next, let's quickly take a look at the theory behind this. So now here's a quiz for you. Suppose we have a district region, and we know I is 3. Now the question is, what are the values of the L, R, and N? So we could choose many different combinations, of course. For example, 113 or so many others. And in fact, we cannot determine exact values because this is under constraint. Actually, this is the case of vision. So in this occasion, L was a luminance, and R was a reflectance, and N was a surface normal, and I was a resultant image. In that sense, image is given by the multiplication of these three factors. So put it a little bit more formal. So light exists everywhere, and they are reflected by a surface, and reflected according to the reflectance properties of the surface normal. And then some of the reflected light fall onto the retina. And what actually we can get from the sensory system is only this retinal image. And to know what an object is, we have to infer R and N. In this way, the problem of vision is an ill-posed problem. And we cannot solve this problem by measuring only the physical properties. And what happens is, in here is that prior probability. So it helps us in solving this problem. So in other words, the prior knowledge about the world. But I will leave this for another time and move on to another challenge in vision. So another problem concerns how the visual system processes images. On the retina, projected image is decomposed into small elements, somewhat similar to pixel in a digital image. And this illustrates how an image is presented by pixels. You can see many numbers each, indicating the brightness at each position.
Before you go, just small question. Do you mean that the retina doesn't send independent information? Only information for images, the luminescence and information about L, R, and N are not sent by retina. Is that what you mean?
What is the last?
So you said so to construct image, you need to know three factors.
Yes.
But retina doesn't send these three factors independently.
No, I don't think so.
But not just the image. Some information about the luminescence might be sent.
You mean a wavelength?
Wavelength, so whatever.
Oh.
Brightness.
Oh, yes, of course. It's the information about the luminance. Yeah, I didn't clearly show that here. Yeah.
And Bayesian computation is of course done by in cortex.
Yes, I think so. Okay. Any questions so far? Okay. Yeah, if you get any questions, please ask me anytime. I think I was here. Okay. Okay, and you can see many numbers, and each indicating the brightness at each position. But of course, only from these numbers, we cannot tell what this image is. And in fact, this was a part of the human face about this part. But remember, the retinal image is all that we get from our sensory system. Then what does the visual system do to see the object? So it gradually groups these local information and construct object and scenes. So the key points are grouping and construction. But you might have not been convinced yet. So let me show a couple demos. So let's begin with another quiz. So this slide shows several identical alphabet characters arranged in a white table. But white ink was spilled over them. So you can see only parts of it. Can you tell which letter they are? Well, this example is quite famous, so some of you may have seen this before. Has anyone seen this before? Okay, very good audience. So maybe you can take a look for a second. Is there anyone find what this is? No. Okay, it's hard to tell. So let me change this white ink into black one, okay? Now you can clearly see they are beads. Okay, let's see. I just want to point out here that because of this contextual difference, because what we see about the beads identical. For example, this part is also shown in here, and so does all of the parts. So B itself is identical. Only the difference is located outside of the B. Okay, and because of this contextual difference, we are able to group parts of each object and construct the perception of the beads. So let's see one more example. So here's another quiz. So this slide shows many line segments, and there is a bar comprised of some line segments. So, can you see a bar? It's very difficult again. So let me give you one hint. The bar is about this size. Can you find it? Okay, this is actually very difficult. Now, let me move this board from the bottom to the top. Okay, ready? Okay, here comes the board. Did you see? Let's do once more again. So here comes board. But when it stops, it's gradually fade out. Okay, did you see the board? Okay, good. So this is just like the previous example. The physical properties of the images are identical, except for the common motion of the board. And this common motion allowed us to group segments and construct the perception of the board. Okay, so I hope many of you have realized that grouping and the constructions are the crucial for the perception of the object. Now I'd like to move on and briefly show what's happening in the visual system. So this slide shows a very simplified version of the ventral visual pathway that contribute to the object recognition. So images projected on the retina go to the backside of the cortex, named visual area V1. Well, actually visual area 1. Sometimes we call the V1. And they gradually move to the front side through intermediate areas like V2, V4 into IT. So let me briefly show what's happening in this visual pathway. So this slide illustrates how images are gradually processed through V1, V2, V4, and to IT. And in this example, printed by Dr. Kuniyoshi, you can see many human bodies arranged to form a single face. And to make a long story very short, V1 neurons represent local contours. Oops, excuse me. Where's my pointer? Yeah, V1 neurons represents the borders of the visual features, such as luminance, color, orientation, and texture. And here, borders are shown in the black line, and some of them are marked by red. And in the next area, V2, neurons represent contours. In V1, sorry, it's very difficult to, oops, sorry, there's my pointer. Okay, sorry. So in V1, neurons code borders, but they don't know which side of the border belongs to an object. For example, this border is a part of the person on the right side, but nothing to do with the left side. So this is the contour of the right side. And to determine the contour from the border, they must know which side of the border belongs to an object. Actually, V2 neurons represent contours that include the side of an object, which is often called border ownership. So let me explain this idea just a little bit. So in this example of a small square, the side of an object's border ownership is obvious, as shown in the red arrows. And so is in this print. For example, this border belongs to the right. So the border ownership is on to the right. And actually, this border ownership was the key for the first quiz. So in this case with white ink, for example, this red border, this belongs to the left, okay? But in this case with black ink, this border belongs to the right. Actually, it was a contour of the black ink, not of V. So in this case with white ink, we perceived incorrect border ownership, which made us to incorrectly group the segment and fail to construct Bs. That was what happened in your brain. And physiologically, Fondelheit and his colleague at the Johns Hopkins reported that many neurons are responsible and selective to this border ownership. And this slide shows the report of a single example neuron. So these are stimuli they show to monkeys, and this ellipse is a classical receptor field where this neuron was looking at. And this bar shows the response of the neuron to the stimuli above. And across all stimuli, stronger responses are shown in the red. And in this pair of stimuli, the response was stronger when the object was on the right. And for example, in this pair of stimuli, again, the response was stronger when object was on the right. So in this way, this neuron was considered selective to border ownership to right. And they show that many neurons are selective to border ownership in V2, or selective to contours. And a later study also clarified the responsive field of these neurons. And this shows the estimated responsive field of 2 example neurons and the mean across many neurons. So we observed facilitatory and the suppressed region in regard to border ownership. And a model based on this responsive field naturally reproduced the border ownership in natural images. And this slide shows example of a simulation result. So yellow shows the correct estimation of border ownership, and blue, incorrect one. And this result shows a good agreement with the human perception. For example, in this closed region, we often see a figure inside a closure, and so this is the model. But it was actually a window, as you can see in this original image. It was a window, so the correct ownership should go to the building, not to the window. But we often see this as a one object, and so this is a model. So I have briefly shown what's happening in the V2. Specifically, contours are constructed based on the estimation of border ownership. Now, I'd like to move on to the next area, V4. Any questions so far? Okay, good. So V4 neurons represent a surface that is the extent of an object, which is often called a figure with respective ground. So let's begin with yet another quiz. So this slide shows a black and white picture that contains a dog. Can you tell where is the dog? Okay, someone did. Nice. So it may be difficult to find it the first time, but once you find it, you see it forever. So if this is the first time you see this, spend a couple seconds to try to find where is the dog. Okay. Has anyone found the dog? Okay, great. Okay, maybe you can tell that. Here is the duck facing downward, and this is the body. Here is the front legs, and this is the hind leg. How do you feel? Did you see the duck? No? Yes? How many of you see the duck? Okay, good. Well, if you didn't, don't worry about it. But
Dog's head is right side, not the left
left side, here is the duck.
Yeah, but
Yeah.
Okay. Anyway, there is another head.
Oh, really? Where did you see?
Right top.
Right top?
Not a little bit. Anyway, yeah, a little bit down.
Down?
That,
Around here? Oh, here.
that look.
Oh, yeah. Probably.
It's the division. Another dog.
So this is Dalmatian, so maybe there's 101.
Yeah, that is, yeah.
Well, anyway, this is quite famous one, so maybe you can find easily on the web. So if you couldn't see it here, try on the web. So anyway, the point is that we have an ability to group elements and construct a surface. And once we know what we need to group. So let me show one more example. So this slide shows a watercolor effect. You may notice that this region looks a little bit orange, and so does this region. Do you see on the screen? Yeah, maybe. Okay, this is, if I erase this contour, of course, it disappears, right? So actually, this is white. But if I put this contour back, you can see a slight orange. And this effect is also called the filling in, because orange on the one side of the border seems to spread into the surface or filling in the surface. Again, the point of this example is we have an ability to construct a surface, which is a spatial extent of an object. And once we perceive the contours. Now the question is, what's happening in the V4? Our hypothesis was that V4 neurons construct surfaces. To test this idea, we need to address two challenges. First, we could not use just a square or circle, but a variety of shapes. Otherwise, we end up with finding the neurons responsive to a circle or square. And second, a familiarity may influence the perception. So if the surface looks like a known object, neural responses may reflect its recognition rather than the surface construction. So we needed the unfamiliar shape. And to address these challenges, we use local patches of natural images. By randomly selecting a small region from a natural image, we generated a variety of unfamiliar stimuli. And this shows a few examples of local patches, those with natural colors and texture and their silhouette. Then we showed hundreds of them to the monkeys and recorded from V4. Again, our hypothesis was that V4 neurons construct surfaces. And to test this idea, we recorded from V4 neurons and compared the responses to figure and ground. And we expect that responses to figure is greater than ground. Oops, sorry. And this slide shows the result of an example cell. And this, sorry. Okay. This icon indicates the responses to a dark figure, assuming that we see a figure in this black shape. And this small red dots show the rest of the field. So actually, cells are looking at this location. And this icon shows the response to dark ground, because white region looks figure and dark looks ground. And these are icons for bright figure and ground respectively. And in this cell, we see the larger responses to figure in this case or this case. So it's independent of contrast. So this result indicates that this V4 neuron is responsive to figure. And this shows some more analysis. This is analysis where this has failed, and these are temporal histograms. And this is another example cell. This cell also shows a stronger responses to figures independent of the contrast. So these are just the two cells. So how about the population? So we tested a few hundred neurons and found that about a quarter of neurons showed significance to figure ground. Now we know that many V4 neurons respond to figure. The next question is how consistently they can tell figure from ground. In other words, what is the correct rate across a wide variety of natural images? So we calculate the mean correct rate across stimuli and neurons. And this slide shows the result. This shows the mean correct rate across individual neurons in the figure ground determination. So with any condition, the correct rate was low, just slightly above the chance rate, which is 50%. So this result indicates that individual neurons cannot reliably tell which region is a figure. So this may not be surprising because we are talking about a single cell. So single cell may not be able to handle a wide variety of figure ground. So if so, the next question is whether a group of neurons is able to tell figure from ground. So we expect population coding of figure in the ground. But we don't know how the responses of a neuron are combined or integrated. So we decided to use SVM classifier. In short, we used machine learning to find the best possible combination of neurons to get figure ground signal. And by using SVM, we combined the response of the neurons and estimated whether they looked at figure or ground. And here's the result. This shows the mean correct rate of the SVM as a function of the number of the cells are combined. As the number of the neurons increases, mean correct rate also increases up to about 80%, depending on the condition. And around the 40 neurons achieved about 70 to 75% correct, indicating that about 40 neurons are able to determine figure ground. Now, what do you think about the 40? Is it larger than expected or smaller? So what I mean here is a pool of the 40 neurons can distinguish figure from ground for any kind of the shapes. So to get the idea, we computed the intrinsic dimensionality. In short, dimensionality is the number of the significant component of PCA, principal component analysis. The results show that dimensionality of V4 neuron to natural image patches was around 40, which agreed with the result of SVM. So this result suggests that figure ground determination may be a principal function of the V4 neurons. So far, I have talked about the correct rate across all stimuli, but the overall correct rate alone does not tell much. So we now think about each stimulus. Now the question is, what stimuli evoked correct responses and what did not? Perhaps the most important criterion here is whether responses agreed with the human perception. So we examine whether stimuli easy for human were also easy for neurons in figure ground determination. So to address this question, first we carried out the human psychophysics. So in short, we showed to human participants the same stimulus as that we showed to the monkeys, and identified the easiness of a stimuli in judging figure ground. Oh, and Here, what I mean by easy is that they can always see the same figure. And if stimulus is difficult, they confuse and see different figure each time. So easiness is the same as the consistency. Okay? So this slide shows the result. We observe a wide range of perceptual consistency between 50 and 100% from chance to perfect. And these are example stimuli easy to judge FG. And these are stimuli difficult to judge. You may be able to feel that some of figures is easily findable, but perhaps in this stimuli, it's difficult to find a figure, or you may see multiple figures. Now, we want to examine whether neurons can easily judge these easy stimuli, but have difficulty with these difficult stimuli. And here's the result. This slide shows a correlation between the perceptual consistency and the signals response. The correlation was significant, but not really apparent. But this result may be expected because single cell corrected was low, it was just above the chance rate, as we earlier. So how about the population? So what we see before was that 40 neurons get about the 75% correct. So we expect that a population of neurons showed a good agreement with human perception. But we don't know, again, how the responses of the neurons are combined. So we use the SVM classifier again. So we pulled neural data, and the machine just a figure ground at each location. In this case, figure is shown in orange and ground in blue. And for the learning, we need a ground truth. So we gave the human judgment of figure ground as a ground truth and let SVM learn how to judge. And for the ground truth, let's see the result. This slide shows two example stimuli. And this is the one example. And this is the result of SVM. The orange shows figure and bluish shows ground. So in this case, I showed the two stimuli, identical stimuli in the opposite contrast. So this is a black figure, white figure, but the machine sells the same region is figure, independent of the contrast. And this is a consistency, population neural consistency. So this indicates how confident the machine was. And this shows another example. As similar to the first one, the machine naturally detects the figure region with some confidence. So now we have the population consistency. So we can compare it with perceptual consistency. And this shows the result. And this shows a correlation between the population neural consistency and human perceptual consistency. We can observe a clear correlation between them. And stimuli easy for human was also easy for machine. And difficult for human was also difficult for machine. And this correlation increases as the number of neurons combined increases up to about 80%. So this result indicates that the response will be for neurons agreed with human perception. And the result we have seen so far suggested that a population will be for neurons construct surfaces. Now I'd like to move on and briefly show what are the cues for this construction. So frankly speaking, we haven't got the conclusion yet. But our recent study showed that multiple features of contours play important roles. And to make a long story short, very short, we estimate the optimal stimuli of FG neurons. We expected that the optimal stimuli must reflect the cues used by the neurons. But how we can estimate the most responsive stimuli of the neuron? This is, there's a bunch of the different images of scenes in the world. And we want to find out which one makes cell the most responsive. Fortunately, one way to do so has been proposed by Ponce et al. Recently. And they showed they use the generative neural networks to generate the optimal stimuli. And to do that, they make a loop with a real neuron. And so here's a loop. They make an optimal stimulus and show to the monkey and get the result. And then again, this network makes a slightly better stimuli. In that way, they get the optimal stimuli. So we wanted to use their method, but this method needs a lot of efforts and time. So we replace this real neuron with a model. Actually, this model consisted of the deep net that reproduces real V4 neurons with about 95% explainable variance. So this method was first proposed by the Bashvan et al. At MIT. So we use it for the model. And we train this network with our neurons. So we applied this generative network to the model neuron and estimated the optimal stimuli of the real neurons. And this shows the estimated optimal stimuli and their responses. The blue dots show the responses to our natural stimuli, and red dot optimal stimuli. And actually, each dot indicate responses to single stimulus, and each column indicated responses to a single neuron. So we can see that the optimal stimuli in red, in fact, evoke the strong responses than what we showed to them. Now, we have the optimal stimuli. So let's take a look what they are. The first thing we noticed was that the optimal stimuli consist of multiple components, specifically multiple coverages, which is evident when comparing them with the natural images. So we examine whether these neurons code multiple component and coverages. And this shows the contribution of the components to the neural responses. Actually, this, you may be able to see some of these curves, and they are a part of the optimal stimuli, which were fallen onto the classical receptive field of this neuron. Okay, so we want to check which part generated stronger responses. And the colors indicate the degree of contribution. The white and black indicating the strong and weak responses, respectively. And in this example neuron, we see that at least 3 components contributed substantially. So how about other neurons? And this shows other example neurons. Those marked by the red was responsive to multiple components. So many neurons showed the contribution of multiple components. And next, we examine the quantitatively. So this slide shows an example neuron that the response actually increases as the number of components increases. So how about the population? So this slide shows how much the response increased. Oops. Oh. When we added one more component with respect to the original. So for example, if we have a 2 component here, we compare the response to those to 1 component, okay? And each dot represents a single neuron. And we can see that most of the neurons lie above diagonal, indicating that these neurons responded more strongly when an additional component was added. So these results indicate that V4 neurons appear to code multiple components and features simultaneously. Today, I didn't have enough time to show what features are coded, but we think that a population of V4 neurons encode multiple components and features and contribute to the surface construction. So today, I have briefly showed what's happening in the middle of the ventral visual pathway. Specifically, V2 neurons represent contours by detecting border ownership, while a population of neurons represents surfaces by detecting clear ground, while simultaneously coding multiple component features. Okay, thank you for listening.
Any questions from the floor? Yeah.
Yeah, please.
Sorry, I couldn't see who asked. No?
I can answer.
Yes.
So just in the middle of the talk, you showed this example with the shape that has a black line and then an orange line inside. And then when you remove one side, then we don't see the surface anymore drawn. So what do you expect in your V4 neuron population when you take this surface and then you just do a slight modification that does not allow to recognize the shape anymore?
Yeah, good question. So we expect that neurons response to fear responds to that orange region if they are looking at that part. But other FG neurons looking at the blue part, they wouldn't respond strongly. That is our expectation. So I think where we see orange, that is that we think the evidence that surface is constructed there, although there's nothing. So it's kind of an illusion. And I expect that V4 neurons respond to that illusion.
And do you think if you were removing little piece by little piece of a contour surface, how will the population behave? Like you will lose neurons, or you will lose an overall correlation between all of them?
Yeah, I think so. If you remove the orange from the contours, then they get confused.
Yeah.
And the response to the previously orange part and the blue part, those will be similar, and we cannot distinguish between them. That means that we can see surface in either side.
Yes.
Thank you. Simple question. You said a quarter of the neurons do figure ground. Is it a quarter of the responding neurons? Or is it a quarter of the whole population? And I'm also wondering, is the code a combinatorial code?
What did you say is the last?
Is the code a combinatorial code, like different surfaces have different combinations of neurons?
Yeah, that's a good question. To answer the first question, yeah, we recorded from a few hundred neurons, and a quarter of them show the significance to the response in the differences between figure and ground. So, and the next part, how they are, what is the populational representation? That is a very important part. Actually, we are now trying to find that, working with Dr. Yukaku Yamane. But at this stage, we have no idea. And the one thing is that I don't have much expectation in that I can show you in this kind of the beautiful slide, because it would be very high dimensional. So even if we, make it low dimensional, still, I'm not sure whether we can get intuitive interpretation on that. Yeah. But basically, that's what we think about it.
Thanks.
Sorry, thank you for the talk. So you mentioned the intrinsic dimensionality was 40. I was wondering, like, is that depend on the complexity of the scene, or is scene independent?
Excuse me, what do you mean?
Are the intrinsic dimensionality, like if I have a very simple like picture, will that?
Okay. Yeah, actually, that dimensionality was computed from the combination of the stimuli and neural responses. Okay, and then yeah, your idea is correct because it was relatively simple. We get a 40, and Sidney Leakey, who used to work for the Salk, he showed that the dimensionality of the IT neuron respond to natural images was about 100. So more complex, we get more, I mean, a higher dimensionality.
We should use mic. I want to ask you about I forgot the English term, perceptual library. So famous example is two persons facing each other. This looks like vessel. And so you record this related, I think, two aspects. One is border ownership in V2, and another is peer background segregation in V4. Because I think both border ownership and peer background relationship changes when perceptual changes. So my question is first, so did you observe any kind of oscillations in V4 neuron that might represent switching in perception?
Yeah, very good question. In V4, I think no one has been done that. But in the V2, the Rudiger Ponderhart group do a little bit similar to that. They observe the oscillation depending on what they show. Yeah.
Maybe you do not know the answer, but I wonder which actually drives, V4 or V2? So if top-down is important, I believe V4 is the driver.
Yeah, that is also a good question.
But can you replicate it? So I guess you make a model. In the model, did you find any oscillatory behavior when stimuli is complicated?
Yeah, actually, one of my former students, now Dr. Wagatsuma, he carried out the simulation and he naturally reproduced the oscillation to the perceptual library of the face and the face. Yeah. But in that model, he used only the V2 neurons. So back to your first question, whether V2 or V4, I'm not sure, but I feel that it's happening in both. Yeah, because if V2 and V4 get the contradictory responses, we may not get the concept of a face of a base, maybe get something others. So I think it's happening at the same time.
Thank you.
Have a little bit of a random question about the connection between this V2 and V4 with the psychiatric disorder. To diagnose the schizophrenia, you show like an ink blot, which is called like Rorschach ink blot test. And then when he or she is having schizophrenia syndrome, and then they see like a dead body or someone's face, a scary face, does it mean that in the schizophrenia patient, V2 or V4 area is affected, or it's more about the cortex computation?
Yeah, frankly speaking, I don't have an answer, but I think it's happening in many parts of the brain simultaneously. And perhaps that the origin of those syndromes are the kind of the changes in the dynamics or the strategy in the brain. So that may affect all of the visual area, B2, B4, and IT, and so on. And yeah, I don't come up with any idea to segregate which part do that. But yeah, that's a very interesting question. Oh.
Maybe mic is on, so you can speak.
Oh.
Okay.
Push.
So in the last part of the presentation, you showed the different parts of the stimulus, which has it's the this one. Do you find any shape parts that reduce the response?
You mean the future of the contour? Yes, actually we examined that, and yes, it turned out that the very curved contour was very effective. The curved contour was effective and contributed a lot, and the straight line, not much.
But reduce, I mean, response.
Inhibitory, yes. That's also a very question. Actually, among the some tens of neurons, we didn't observe such inhibition. In here, we see only the single inhibitory one. So in this guy only, this single cell showed what you said. But all other, if we increase the whatever the component is, we get the increased response.
It means that most of the cases that the response is additive.
Yes, yeah, that was not expected, but so far, this is what we found.
Any other questions? Maybe it's time to close. So let's thank Dr. Sakai again.